Okay.
Yeah, so hi everyone. Thank you for joining this discussion. So this is our week 10, and hope you goes well with our lecture 10. So this week our discussion topic is related to the FVCOM based ecosystem models. So if you have any questions regarding to this lecture, you may read them up. Or after this section, if you have any other questions regarding FACOM, so you may also ask the Dr. Chen. So anyone have any questions?
Um, I want to ask some question about
Yes, please.
about this lecture. Yeah. I share my screen.
Yes, please go ahead.
Okay, in the lecture, actually, I didn't understand fully because ecosystem is too difficult for me. But lecture said FBCOM output are forcing the ecosystem model, and in my understanding, is forcing is one-way direction from FBCOM to ecosystem model. And in the lecture, there was a coupled case with FBCOM and the ERSEM, and I want to know do the biological variable calculated by ecosystem model directly influence to physical ocean condition? And if the feedback is relatively small, I think coupling offline simulation will be more good to save some calculate cost. Yeah. So I want to know, is there any modeling case which show the ecological process leads some noticeable change in physical condition?
Okay.
Thank you. Dr. Chen, so this week, shall we still keep
We
the...
still do like a Professor Wu suggests, we
Okay.
get all the questions and we answer at one time. That's why we don't have like one question for all time. That's why we understand more opportunity. Okay. Yeah, just the PowerPoint here, so we can ask another question, another one.
Yes.
Kind of one.
Okay, I will show a question. So I share the screen. So my question is here. So in slide 4, the phytoplankton calculated for this equation, and in mu sub I is calculated to two equations for A and B, and A is considered the factor to minimize the growth rate, and B is consider all factors to affect to growth rate, and in FFCOM jam is considered together, and it has weight factor in alpha. So how we consider the alpha factor to calculate is have any other know-how is my question.
Okay, good.
Okay, thank you, Jamil. So next one.
Oh, I have a question. Can I share
Yes,
my
please.
screen?
Yes, please.
I will. Okay. Oh, okay. Oh, is it is similar to Jimyoung's question? In this week's class, I learned about the ecosystem model. Among the topics, I became curious about the equation for calculating the gross growth rate of the phytoplankton. I understand that the gross growth rate is determined by light intensity, water temperature, and limiting nutrients. But why is the equation split into the two parts and then calculated by applying weight to both equations? I think it might be to assign different weight to the effect of nutrient and light intensity on the growth rate for each phytoplankton species. Is my understanding correct? Yes. Okay.
Okay, thank you. Next one.
형아, 마이크 안 들려.
I have a question. Can I share my screen?
Yes,
Yeah.
please.
In the class, it was explained that FVCOM gem has a total of 11 light limitation functions. Given that Gyeonggi Bay has a large tidal range and shallow water depths, I'm wondering which light limitation function would be most appropriate to choose for this environment. This is my question.
Okay.
Okay, thank you.
Do you have more? If you don't have more, we can go start from first one. Can you put the PowerPoint out again? I can explain to you. Okay, first one.
I think I can present it here.
Good.
Okay, Yunji, thank you.
No, I think it's better for Dr. Lee share the screen because when Professor Chen need to write something, is it matter whether
Yeah. Okay.
doesn't matter?
Okay, Dr. Chen, I can still write something now.
Okay.
Okay.
So, now go back to slide 2. Okay. Okay, it will shut down.
Okay, so I will share.
Okay. So, first one. So, so you look at this is structures. You can see that the model. Let me put this one on, so I can make the module easily. Okay, that's good. So, you look about the force equation here. So you don't worry about the physical forcing here. That's the forcing just for FVCOM. Okay, so your FVCOM will run, you just keep running, but only difference like a light temperature function, this advection, the intensity, that's called physical part. Temperature, that's the input, but that's directly come from FVCOM model. Okay, that's why, but then if we get a 1D example, it teach you how to build a model because, you can select how many nutrients, how many phytoplankton, zooplankton over there, okay? So you can choose a variable. So they have some special function over there. You can build that. You also can put your own one in if you want to add something, okay? So the whole structure for this generation generates the biological module, which is provide you a platform to build your own model. So you can, for example, you can choose like a nutrient. You can choose one nutrient, nitrite. You also can choose like a ammonia for couple one, depending on how many you want. So for the phytoplankton, you can choose one species. Then you just have a total phytoplankton biomass, or you can choose the multi, like a small phytoplankton, larger phytoplankton, or you can choose a different type of phytoplankton. Right? So we give it a 1D example to show you how to do that, right? So the problem, so the issue is here, so you can run the model as as the online. You also can run the offline. So most time people run the offline, don't run online. Okay, the reason very simple, because when you do the physical model, you probably need to do the data assimilation. Right? Because you got some data in there, for example, ST data simulation, make sure you physically, the model can be provided most optimal fields to drive a biological model. So this way, but if you run online, so you are very slow because you're more the simulation, very complicated. Most online people, when do online, they don't do the data simulation, right? But most of the application you want to get a correct physical field, you mostly do the simulation. Then after this one, you should use the kind of offline model. So then that's the one thing. You look at second of slides, you get another slice, another model. So this is, that's a complete different model. No. Just the same slice.
No. Second page.
Yeah, second case.
Second
Okay.
page.
Yeah, second page. You can see this model is different. These sort of couple mean sometimes you can change the words. It's not a really fully couple. This Earthen and FVCOM can run online. Okay, that's the same as the general biological module. It's no problem. But most people don't use that, still use the offline, like I said. But this model different from the general biological model. This include a geochemical process. The FVCOM model in FVCOM code, this coding only can do the low track food web, but not consider biogeochemistry, that process. So that's model not able to predict the CO2, right? If you go there, they assume when they use like no FVCOM general biological model, so they assume that CO2 is available, very sufficient for the photosynthesis for other, but they don't do the CO2. But this one can simulation the PCO2, tell you, okay? PCO2 also can teach you pH. That's why this model can use for ocean acidification study, but another one cannot. Okay, that's the difference. Okay, you look at the model here, use a pen. You see this part is completely this part, okay, right? You see this part different from another model. This part is a biogeochemistry process. This part, that's the basic process. Okay, then you look at the genetic biological model, that's the most watercolor model. No basic, no biogeochemistry process. Okay, if you want to study climate change impact, that model not able to do that. Okay, that's just typically like what we call MPZD type of model. Okay, that's only to do the low traffic food web. But this model include water column ecosystem like the other one, but difference another one you could build by yourself, right? This one is fix it. They only consider 4 of a phytoplankton, make a figure for all other inorganic organic like a carbon for others. Okay, but important things, the difference is this model 4 things include biogeochemistry process. Another second include basic. Okay, you can include the basic biogeochemical process, also food web was in basic. So that the difference is what really right now. People, when you do the testing for the column, because some regions the basic are not very important, you use the previous model, so you can get a whole simulation. You, for example, you put a nutrient in, right? You run a couple years, they get a conservative, seeing the cycle come out. But this one's different. This one, because when you, for example, phytoplankton die, zooplankton die, they go to the basic first. Then after this one, there was regeneration nutrient back from basic flux into the water column. So this one is not like a linear system, just a nonlinear system. So that's why you be carefully choose the transition, make sure they repeat your season cycle, right? But the previous one, they definitely repeat because that's a conservative model. So if a nutrient die, sorry, if a phytoplankton die, zooplankton die, a grazing rate make the zooplankton lose. But all these things in the water column, intermediate decomposition, go back to the new nutrient. As a system is conservative, but this one they go to basic first, then basic come out again. But this system is not linear. Sometimes you cannot get a conservative run for for resolve system evaluation. That's why you have to be careful to choose parameterization. I has been talked to the model development last November last October when I was UK. So I asked him to add like a two loop. One is basic, one is you don't need to go to basic. You can direct get a loop, loop in the water column. Okay. So they said, oh, that's I showed them what the example is. The problem is, so then they said they want to do that. But then last couple of weeks, I found the person developed the model left UK. They're working for consulting company in Spain right now. So that's why, but this model is pretty good. No, the developer told me they already update the new coding before he leave, so can run. They get a lot some kind of improvement. I don't know what improvement they did, but we also do a lot of improvement too. We get a conservation improvement, so we try to get together. That's the way. But the question you have one is that if you had a offline, you want to see that have people this kind of feedback. Yes, they do have a study about the feedback. I couldn't remember exactly what time. It's like a long time ago. Most of the feedback is changing light. No, because you had a phytoplankton bloom happen in the near surface, the light penetration can be changes, right? Before is for example, water is clean, so light can go through easily, right? But light penetration changes, so your photic layer can be changes depth. Your photic layer just like a photosensor layer, right? In the surface can be changes. So in this way, okay, then you can see, so these things like photosynthesis in the near surface can be changed in the up layer. But then the person will study very hard. They're gonna model it. He published a paper in the JG. I couldn't remember what the time is. Then he found feedback after changing light, the phytoplankton can change based on observation data here. He said, no, some kind of simple model. He said only about 1 or 2%.
I'm sorry?
No, just a feedback effect by the phytoplankton to light, right? The changing light, then after changing light, nearby the front zone. So the feedback, how the biomass changes, the growing rate or other change rate, about 1 or 2%. Very small.
Okay.
So this way, so that's the reason most people don't consider feedback. Okay. The most of the feedback is just the light, because your light also had a pen. No, the problem is biological model most called the parameterization model. They got a lot of parameterizers driving the model. Okay, for example, you get a 2 or 3% feedback. They can lead between about the parameter. They can count it for that.
Right.
It's a problem. It's a biological model, not like a physical model. Everything's solid except the mixing. Except the vertical mixing, horizontal mixing. That's the two things we have to consider. Kind of wind stress and some kind of polarization. We had a lot of reason to make the parameter changes. But biological model, everything is the parameter. For example, you talk about growth rate, so the growth rate had a range 0.5 to 1.5, right? So because they don't have a lot of observation, right? They don't have a solid, because it's kind of like some phytoplankton like a diatom. The growth rate of Georgia Bank and the growth rate in South Korea could be different. Right? That's why you don't have a very solid, numbers here. That's exactly growth rate you have. You have a range, you can tune the model. So that's why a lot of, like a mathematician, like a physical person, don't trust the ecosystem model. Your ecosystem model just put every parameterization together. So much physical, you can tune the number, get everything you want. That's partially true. Okay. However, that's why for all ecosystem study, you have to do the sensitivity study. Okay, because you get a lot of the kind of, number, kind of constant number, the closing number, this kind of parameter. You have to make sure which parameter is more sensitive to your result. So you will tell people, okay, so that so you tell people this one. So this one, that's the most model uncertainty come from this parameter, right? But that's the way people do. Okay, so yeah.
Professor Chen, I mean, from Yunji's question, so in this paper, for the coupling, is it necessary to study acidification, something like that? But if that is not our major interest, then is it also possible to run RSM model separately offline?
Oh, most everyone run Earth model separately. No one run online. Okay.
Okay, I see.
Okay. So
For this study, for this paper.
Yeah, for the paper, this paper is also kind of offline.
Oh.
But he said a couple. Couple has a lot of meaning.
Oh, I'm sorry. So I mean, let me correct my question. So, is it necessary to run RSM coupled? Is it also possible to run RSM model uncoupled separately?
Yeah, that's exactly can run separately.
Mm.
So this way, you have a FVCOM run, for example, you run a FVCOM for 1 year, right? You get all the hourly output FVCOM result there. So you can use the FVCOM hourly drive the person.
Mm.
Okay, you don't need a couple. Okay, that's what I want to tell you. Most people don't use a couple. For example, this paper done by Dr. Wang, he run for a whole couple year for physical model, right? So then he used the physical fair to drive it. You can do the data simulation, input the physical fair. After that's why he just run the Earth separately, but with the FVCOM as the input file. Okay,
So
but if you have FVCOM, you have a net CD5 there, you can direct the code FVCOM, they can input data to your model.
actually, Incheon City, the local city, really wants to develop this RCM type of model near Gyeonggi region. And the question, as Professor Chen's, I mean, as you mentioned, because there are so many parameters,
Yeah.
so many factors, is there any kind of a way to set up systematically? And you said it's a sensitive test. Yeah, but so many parameters. Which parameter sensitive test they should do first?
No. This way, most people most sensitive the parameter you can see, is a closing rate, like a mortality rate. Also, you can see this kind of grazing rate. So this is parameters most of the more key important. That's why like Dr. Wang did this one. He used he mostly had 200 parameter. Okay, then he choose the 1% changes of all parameter, though each one he run the model, so can see that the results, can how much change for result. Like if you look at my previous paper, I described how the method is. So then if we get a 5% result change, that means the parameter is sensitive. If not, just an insensitive parameter. So you can choose like a that's the reason a lot of people don't want to use a too complicated model. They go back and use like a previous page, like an MPCT model type, because this model parameter is still a lot, but much less, right? Because you can look at this parameter, you get a basic parameter, you get a reward column parameter, you get a main. But when most people apply the model, the first thing is they just use the default value. Because the model tell you default value. They get each range default value. You can run the model, see how the performance is. So then after this one, you want to see, for example, you get a phytoplankton lower, than observation. Also, you get some, some parameter like a PCO2 or something lower. So then you can think about what kind of biological physical process for observation for all other study. So you can just tune some number, the parameter for this component. You can adjust results, make it like a compare observation. The beauty is, after adjustment, you cannot keep in changes. Okay, if you keep in changes, if for example you run 1 month, the parameter next month change another parameter, then the results are non-useful. But most times just change it 1 time, 2 times, but you keep the constant number, you can see how the performance they are. Okay, but so you have to practice to learn how sensitive you are. That's why we provide a 1D case, let you test it, let you play. Okay, see how you can see you change your parameter, the results can change it quickly. Okay, right? But you can practice that.
Right.
No, your question is that if so many parameter, how you set the parameter? Most times it's model give you default setup. Okay, you just run a default setup. So later you adjust some key parameter. Okay.
I see.
Okay. Okay. Yeah. Go to another question. Another question here. That's a good question. Okay. As I tell you. No, I teach the course this semester for ecosystem model. So we have to mention, how this kind of, growth rate be defined. That method used here, come from Harvard University. They developed a method. We just adopted. But most of the biological model don't use this method. Okay, use other methods. There's so many different kind of methods. Okay, but then the question come out. So for example, they have called this method just choose if you get n nutrients, they choose the minimum. For example, the A, right? The A just choose the minimum. Okay, but sometimes a lot of per model they don't choose the the second method just put the light function out, right? Special light function, but then you because this all factor is just because like between 0 to 1. Right? They put a light function, they have a different way. So this all basis not based on the science. Okay? No, based on the, on the, on the people's empirical thinking. Right? For example, if you look at the other model, they can put the nutrients all factor times together. Times together.
I'm
because
sorry.
no,
Give me again. What?
Okay, let me show you. Okay. You see, for example, the male I, all right? They use the factor like something like this, nutrient I, okay, half century ratio, then the nutrient I here, okay, all right? So this one choose the minimum. Some people model just times together, like I1 times I2, okay, times I3. Okay, many methods to do that, okay? But this way, you can see this one here. You see this one include the light, it could not include the light here. It's not much different. Most people not choose the average, okay? They just choose the like either the alpha equal to 1 equal 0 to 0. Okay? But that's not the only two methods you can use. You can choose any method you want. No matter, I can tell you the more than 10 methods, okay? Right? I don't show the screen here. I can show you what the slides I have here. So explain what the physics about this one. Okay, if probably I can show the slides to you. Okay, if you're gonna show slides to you, I can show you something. Okay, I can show you my class I teaching. Okay, something, I can show you what I teach you the method, like okay, you see this spring I teach in the course. I think so. So
I will stop on my side.
yeah, what was I had not started. Okay, yeah. Okay, you stop sharing. I sharing the file.
Yes, now we can see your screen.
You can see here. Okay, this lecture show you what the science. Okay, how to do how to define the light, what the function is. But I just show you, I show you the one slide. Okay, see here. You see, that's the measure. For example, kind of uptaking rate, the closing rate, uptaking rate. You see this measure kind of time together, right? So then you have a minimum. You see, then I tell you what the problem if you time together, right? So if you have one nutrient, for example, equal to 1, right? But how we time together the 3, this becomes smaller. 10, you can make much smaller. So some other method look like one, you show this one, they use the minimum because they only want to see which is the smaller one, limited, because it's a nutrient control, nutrient limit, right? So they want to just say that I just want to find a key, okay, nutrient that's a limit of closing. I don't care for others. But this one, time together, they will think of all the nutrients as equally important. Okay, that's why how the closing rate affected depends on the product, what the time together. That's treated as the equally way for all the nutrient. No, but this way said no. This all the closing only depend on the one is really limited, smallest one. You understand this one? Most people like to this way. Most people like to use this way. Okay. The reason because it's make sense. For example, in the most estuary is a phosphate limit system. Most ocean is a nutrient limit system. In the ocean, this part of component important. In the estuary, this part of component important. So I use this one. You can use the minimum one to control system, right? So that I can stop here. So I can you can go back slides.
Isn't it phosphorus always the limiting factor?
Sorry, limiting factor. Sorry.
I mean, the phosphorus is usually the limiting factor for.
Phosphorus only had the limiting factor was for estuary, most estuary shallow water, but not for ocean.
I see.
Okay. No, if you want to really learn the model, you have to take my course. Marine ecosystem course show you, for example, here, if you apply the model to the equatorial region, so Southern Ocean, you have to consider I'm limit. Because you look at the equator, you go to the near the equatorial region, you find the nutrients very high, upwelling, but the phytoplankton is very low in that region because they lack of the iron. The Southern Ocean same way. Okay, that's why you have to leave the change the model, But this way you see there's a minimum here, but most people don't consider, you see that you put the light in, light off, but all the people, most people you use the B, the mu B, okay, but very few people put the light and limit. The limit only choose for nutrient limit. They say the light is a penetration, the closing rate affected, right? So if light is so strongly, like a later the question is sure, light function, so they can be habitat that's a closing. That's why the light and the temperature is usually be a factor there separated, but this only to minimum of it's kind of nutrient, selected a minimum nutrient, okay, for limited. But this one put the light into there, if some phytoplankton, if I'm a lecture sure, the lot for closing just only depend on light. Okay, that's why they put light also has a limited function. That's why they put it into there. But most of the cases you choose a B enough. That's why you choose a B. B also there. There's a B here. You can choose 0, alpha equal to 0. Choose a B, then take the A out. Okay, but so you ask me how to choose a parameterization, but this parameterization, so so depend on how you weight, okay, which is a really critical limit, right? If you think that the light is a really critical limit, you can go to, can put more weight on the A. If you think a nutrients are most limited, right, you can put it in the B.
Mm.
Okay? Understand?
Yeah, I see.
Okay. So then that's what, okay, go to the next one. Go to the next one, yeah.
This is a question from Mingwu Pang.
Yeah. So can you show that? Can you
Uh,
show PowerPoint?
oh, I shared. I will share it again.
Yeah, you should have PowerPoint.
Can you see it now?
No, no, it was no, wait. Yeah. Your understanding, like I think I already answered the question. You see why the difference, why is light put outside, why is nutrient, yes, the why the put light and the nutrient together and the A, why they put the light was outside, only for the nutrient. That's just I answer the question. It depends on how you weigh, see what is really limited nutrient. That's a nutrient limited. That's why you choose minimal, okay, to control the growing rate. Okay, so that's just I think I answered the question. If you don't see the, if you still have come, you can, no, your understanding is is very closed. Okay.
So Professor Chen, I mean, in practically, after, so we can choose our alpha value by trial and error.
No, you think it's most of the time people use alpha equal to 0. They'll use the second one, B. But B's one is much makes sense, right? Because they think of the light have limited, it's mu I and the capital I, its maximum is 1. The value is 1, okay? That's why you can choose like a next question talk about different light function, but most time, like depending on what the region. So if you go to a nearby equatorial region, light is critically important because the light so strongly they find the not grow. Okay, right? So this way you can choose, but most like in our region, just like a mid latitude region, so this way light is most not very strongly, right? So you don't need to pay a lot of attention to light. So you just make sure light, the vertical structure, the effect of vertical structure is fine because most surface light you can mostly put a 1. Okay, in this way the second one is more important because you only have the nutrient limit. You need to find the small nutrients as to control your growing.
If we choose alpha 1, then mu IB
If
is
you choose alpha 1, so then all the function depend on the question 1.
yeah, A.
Yeah, but then this means the really control the new phytoplankton group, both nutrient and also light is compatible, right? So then you probably have light, for example, if you choose if you choose some regions very cold, right? You go to Arctic Ocean, for example. So light is very critical. You don't have enough light on the globe. This probably becomes very important. But if you go to South Korea region, like China Sea region, that's A is not very important. You should choose the B.
And
Okay.
we should not choose B.
Yeah, depending on situation, your light really limit the function now. If your light is really limited function, this also sometimes even you have a lot of nutrients, but you don't have enough light. They're not a glue, right? That means your light is really critical function. Then you choose the A, right? But however, if light is normal, generally, it's really preferred for you by the group. It's not really stronger control. This way, you better choose the B because B just a nutrient limit, right? Now, some model like I did it before, we even don't choose the minimal, we just select one new nutrient. Okay, a limit. So also some other model you can see they have a nutrient limit. For example, equatorial region, they also have a, they also have ammonia control. Because some of the species there, they could be a lot of things based on observation. So even the nutrients small, limited, but they don't have enough ammonia. This phytoplankton not grow. That's why you can put ammonia times as a factor control by the nutrient. You get a lot of function. Okay. I don't
So,
get time to talk to a lot of things here because there's a lot of course study. Okay. Yeah.
so Professor Chen, so I mean, in short, so mu B, the mu IB, the light is treated as more important factor.
No, no, it's a this if you use the B, okay, this means the light function is no, it's not is compatible. No, they're equally, no, it should be equally compatible with the nutrients, but they not compete with the nutrients as which one is really limited to grow. Because if you put a BA, okay, you easily be light control easily by new nutrient control, right? They only choose the minimum one. If light the minimum, they choose the light. They don't care the nutrient anymore. But this one, this light is, normally, okay, equivalent for the nutrient. Now, nutrient is one factor, light also another factor. They equally determined, okay, but not compete each other for limiting.
I see.
Understand?
I see. Yeah.
Yeah.
Okay.
Okay. Most people like we choose the B. Okay.
Yeah. Because we are studying estuary.
Yeah.
Yeah.
Okay.
Okay.
Okay. Good. So another one.
This one is from Yong Song Jun.
Yeah. So you can show the PowerPoint. Okay. Much easier for me. Look at the PowerPoint and explain to you.
Can you see my screen?
No.
Oh.
Yeah, that's good. So I would suggest you keep some parameters, draw all the picture. Okay, no, except the most people use default one is the first one, and only assume light function was potentially decay with vertically. Okay, because surface is more light. Okay, the dip is no light, but that's one people use. But then you can see the steel one. So they use, you see this one, they don't use the light. They just show the surface is one. Okay, surface in the surface is perfect. Okay, then the light function will become reduced. The vertical exponentially vertical becomes smaller. Okay, so this is really match with the light function in the water column, right? So this one, this way. So if you look about sphere drop theory, about euphotic layer definition, that's really close to this one, right? So that's why you didn't see any light on there, right? Most critical important is kind of attenuation factor K. How penetration water depth you have in the water column. The dirty water and the K can be larger. The clean water can be smaller, right? But the second equation still, then you probably don't know the still. How many people understand still? Still is a director in the Wusuo Oceanography Institution before. Very kind person, very nice person. He's my friend. But he's if he's still alive, he probably 100 years old. Okay, he 1996 too. He's the first person to the ocean modeling. He was thinking about the light, the function over here. So they just had limiting. He thinking when the light go to certain regions, okay, so closing cannot continue, grow, right? He had to stop. Okay, that's why they have called limited by light, in tendency. This equation, if you draw the picture there, if you draw the picture, this only, you see this I here, I is equal to this. Okay, that's why you can see, you can draw the different functions there. You can see the there were only two type of function. One is, light was limited, okay, had optic light. Another one just generally not optic light, but only vertical function. You can see this all different function people use for different ocean. Okay, if you draw the picture there, you didn't find much different. Okay, I just asked students to draw the picture. They showed the picture. They showed what happened. It's very similar. I said, which one you should use, right? So I was thinking about, you just go to test it by yourself, because I cannot tell you which one is really difference is smaller. Okay, but I cannot teach you which one is right. Like for example, the one we use in the Massachusetts Bay, we have observation data. Now, have you heard about the PAR measurement?
Mm-hmm.
No, PAR measurement.
Mm-hmm.
Okay, just like a photosynthesis, active photosynthesis, light.
Mm-hmm.
Because light, not all the light can be used for photosynthesis. They only have a certain wave number, like 470, around 470. That's a long way, referred light. They can use the photosynthesis, but they have an instrument to measure its power. Okay, just a photosynthesis active light, light. So use this number put in the model. But the problem is most time you don't have this kind of measure, you don't do the measurement. That's why we had a formula to show you how to convert for the light, for the physical light, come from your satellite data, or come from the satellite measurement, short wave. Use short wave how to convert to, kind of light used for photosynthesis light. Okay, so this way you can use observation. If you do the math, you can see which function really match your equation, your observation. Then you put this one into your model. Okay, so I can show you. That's why beware. You're not able to directly use the shorter wave light into the model. Okay, you cannot do this way. Okay, so if you give me short screen, I can show you.
Yeah.
Okay. So you can see this one here. You see this one here? No, you have a, okay. No, you have a, no, you have a part data here. It's the only light that use the photosynthesis is around here.
Mm-hmm.
Okay, this light, okay, or like this one here, not for, like a ultraviolet light, okay? That's why you see that, so most way, way for the short wave measurement, they include all spectrum. But only the radiation, okay, only radiation about a wavelength of the 400 to 700 can be used for photosynthesis. Right? So that's why we call this one, called this one called a photosynthesis active, okay, radiation. But you have to go to measure. For example, we do the Georgia Bank government region, we do to measure this data. Based on this data, you can see what kind of light function you choose. Okay, from observation. But then the question sometimes, most time you don't have the measurement. So can you use like a short wave from your observation satellite? Right? Can you convert it as the photosynthesis light? So then they had some way to from PAR data, how to convert short wave data. So then we found the most favorable is this one. I should show the equation. Is the most factor there, PAR data and the radiation that you can convert each other. So you can use this one. We found it's the most like a I remember I told you here it's most like a 40%, something like this. So then you can convert the data you see here. this is a PAR data
Mm,
from remote sensing.
mm,
This is a short wave
mm,
data, right? You can convert, but this one you use the model, but not this one.
mm,
So you can based on convert, you can have a function convert from this one times the factor times this one, then put in the model. But this
mm,
really one used for photosynthesis.
mm,
Okay,
mm,
you see, Georgia Bank interior is a different rate.
mm, mm.
Okay,
But
understand?
but it seems it's not really that. Is that really different between Georgia Bank and Gulf of Maine?
No, no, it's a little bit different, not a strong difference. You see here, you see here, you see this one reached to here, this one is 320, but this one almost less than 300. The pattern looks similar, but availability is different.
Yeah, I see.
Okay, you still need to convert.
Mm, mm,
Okay, after convert, you can use it for model.
mm, mm.
Okay, no, you know, if you take my course, you can know how to do that. But you know, for FVCOM modeling, I just teach you about the model, introduce how to use the model, but I don't teach you the dynamics, right? But this course teach you dynamics, why you do this way, why you don't do it another way, how you do that. It's a lot of detail, how you convert the light into the the PAR data, and how speculation, how scientific study. We talk a lot of it. We also tell you how to set the ecosystem model. You know, the water dynamics important. That's something. But this is just for user. Okay, to show you how to use that. Okay. Okay. Other question? Do
So
I answer all question?
Yes, all the four has been answered.
Good.
Is there any other question?
yeah, Professor Cha, I have one question. So to set up this ecological model in an estuary like Incheon, we might, I mean, as a modeler, we might suggest what kind of parameters should be monitored to set up this kind of model. Would you suggest which one is the most important factor if the funding source is kind of limited?
Some parameters really hard to measure. For example, mortality rate.
Mm,
Right? Because phytoplankton mortality, zooplankton mortality, really hard to measure.
mm,
I think it's the four things I would suggest to measure. Just get the PAR measurement.
mm,
The light measurement, your PAR measurement, you can make it easy because your X-ray so changes so great. Your different location, the PAR could be different.
mm,
Right? Some region probably clean water, some region dirty water. That's why you get PAR measurements. So this way, for you at least you get the light more really to the observation. This way you can more result a special difference for this kind of growing rate.
mm,
Right? So then you can see that some people have a method, they go to measure the phytoplankton, like different species. Some people did, like phytoplankton biomass, they compare with the different season. Then you use this way, reverse the calculation what the growing rate should be.
mm, mm,
Right? But this way, this way, who knows correctly, but because a lot of things that can be making the phytoplankton die, right? But then the question about, because you use the one to measure another one to calculate growth rate, but you didn't consider how the mortality rate. Because something, for example, growing, right? Because for example, recently they found, phytoplankton like some diatom, like something. There was a summertime, the wintertime growth is different. We put the growth rate in the model just for constant. I determine no matter summer, winter, right?
Yes.
Right? But all the mortality is constant. Not constant, just like this is some interaction, something, okay? But have a different way to put the mortality. But the question is that some people measure the summer and the winter different, right? But how they determine different, I don't know the biology, how they did that. But you put the model, you get a more beautiful result, right? But you need to do the measurement. We have a program called LTR program. A lot of biological scientists are working on this. Okay, that's why you see that which parameter you can measure is hard to measure. A lot of parameters they measures, they make basis to get a sampling, go back to the laboratory.
Yeah, right.
Right? Get the parameter like a growth rate, they're not able to measure the water. They was go put it back no laboratory to test it. How will see how much it grew?
Yeah. But
You know?
that'll be different in the real in situ situation, right?
Yeah, that's why you have to testing, just use the default value, put it in there, test the model, then you find out which model is more sensitive, the parameter. Then you can tell observation people say, could you mind to do that? To talk to the biology people,
Mm.
okay. Rather than to pay attention, they probably have another smart way to do this, Like we do the same way we do the ecosystem of the Northeast region. We talk about the zooplankton people and the biological people. They say, oh, we know how the way to calculate it. So that's why sometimes they give the growth rate. They say that's the rate that come from observation, right? But sometimes we put the model that look like good, sometimes not good.
Okay.
There's so many parameters.
Yeah.
Okay.
The more parameter, the better.
No. Okay, this is why I teach my class. Depend on comfort level.
Mm.
If you have a whole ecosystem model look like a polynomial fitting function. Polynomial fixing the function. If you consider linear function, you have A and B, 2 parameters.
Yeah.
But you come on a straight line.
Right.
Right? Straight line. But then the comfort level is 2. Then the comfort level high. But then degree freedom 2. That's statistically more meaning sense. Linear function. If you go to hyperbolic, you get a second order term. You probably have 5 parameters. So then your degree of freedom numbers becomes larger. You get a better fitting, but the comfort level drop down. Right? Because the comfort level, then you get a 200 parameter. So that means you have 200 degree freedom. So most results you get has no comfort level. That's what I was at MIT. I'm working with some kind of mathematician, the physical. These people say the term. They never know ecosystem model, okay? They just a mathematician. They said the whole ocean science ecosystem model is garbage. Right? The reason because you get more than 200 degree freedom. You have no kind of statistical meaning anymore. You don't have to come because the comfort level drop down as the degree freedom number increase. Statistical meaning. So that's why we call it ecosystem model called empirical model based on knowledge. Okay. Do I answer your question?
So meaning that if we want to study kind of a primitive study in this region, for the ancient region, maybe it's even better to have small parameters at the beginning.
Yeah, as simple model as you can.
Okay.
Okay, that's why we use the NPZD model.
Okay.
Only consider one phytoplankton,
Yes.
one nutrient. That's why at least the degree of freedom is high.
Okay.
The NPZ model, even don't have detritus, okay, they have 7 parameter only. That's why your degree of freedom is smaller, your comfort level get high.
I see.
But however, biological people don't care that.
Yeah, I see.
For the mathematician, for the modelers, you have to worry about the degree of freedom, you've got the comfort level. But biological people, they don't care. They said, no, oh, you're missing this process, you're missing, detritus is very important. Okay, so then we said bacteria are all important. But you put it, they're all important together, you get a model complicated. We had the people when the ecosystem model started before. I think I'm the one or two couple of people start ecosystem model. Peter Frank and I, and also another professor called Power from Berkeley. Okay, we had a meeting, discuss at the time, nobody do the ocean model, nobody do ecosystem model. So we started doing that. So then we talk a lot about this. But problem is these people retire also pass away. So now the young generation come here, they don't care this anymore. Okay, you told them you don't want to use a complicated model, it's simple. They say, oh, you're missing this, missing that. Right? That's the way. You know,
Okay.
that's the way, like we last time we talked about seminar model. Seminar model is the most difficult problem in the modeling.
Seminar model?
Seminar model.
Sediment. Okay. Yeah.
Right?
Yeah.
So particularly concave sediment, you don't really know what's going on. Like an ecosystem model. That's why I know a couple of professors very famous. They spend all time working sediment model. This whole career get nothing, just like you study a turbulence. Because so many uncertainty.
Yes.
Right?
Right. But practically, the sediment dynamics and ecological things is the thing that people wants to know.
That's exactly.
Right.
That's why they get a very high priority for application, for econ, for fishery, for other. That's why people still use the way like we do the ecosystem model, the empirical method or other method, but at least they can do some better than nothing.
Yes, exactly.
Right? Okay.
Okay.
Yeah, okay. Sorry, get a time. We need to get off. Other question from other people? No question. Let me stop here. No, I get back to Dr. Lee, okay?
Yeah, thank you so much, Dr. Shen, for all the answers for the questions. We really appreciate that. So if there is no,
Next time we ask Dr. Lee to answer the question, okay?
oh, I will try my best. I need your backup. Well, thank you so much for joining this discussion. We really appreciate it, and hope you have a good week, and see you next week. Bye-bye.
Oh, by the way, Dr. Lee, about the
Yes.
homework, any kind of guideline about the deadline of the homework?
Actually, no. So for this online course, because it is non-credit, so there is no deadline for each homework. But for students, if there is any questions, so please feel free to contact us. For example, 2 weeks ago, Yunji sent me some 3 questions about the compilation of the homework 3, homework 5. So I think all of them are good questions, and I solved them. So anyone with any questions, please feel free to contact us.
Great.
For the credit course, we do require to submit the homework at a certain time because we need to get grade. But this course is no grade, so that's why we even I put a time there. So what time the due date, or like this way, but we don't really require.
Okay.
So
But
you
if
can do it the best way. If you don't do it, it's fine.
okay.
But if
But if
you
you have
fine. Okay?
if students have some questions about practicing this example, then we might contact Dr.
Yeah,
Lee.
also we can talk to the class.
Yes.
Yeah,
Great. Okay.
Okay. Thank you so much. So see you next week. Bye-bye.
Yeah.